Hello, I'm Vivian Fregoso, and I'm getting ready to go ahead and pour into the agar dishes. So I'm going to go ahead and melt it. So I'm going to walk you through the process of how to melt this so that we can pour it. Okay, so first we're going to go ahead and put a pot of water, and inside the water we have the holder that's going to hold these in place, a test holder. We are going to put them in, but we're not going to take them off of the we're not going to take the lid off. We're actually going to put them on, but like very loose, just to give it a chance to escape heat. And we're going to do that to all three, and we'll kind of see which ones if we don't need all of them, then we could always put it back in the fridge. And you know how this is a little bit cloudy, we want it to be clear, and that'll indicate to us that it is completely melted. So I'm going to go ahead and leave this to boil. Once it boils, I'm going to get some oven mitts and I'll take them out, and then we'll walk you through the pouring process. So I brought my water up to a boil. So I went ahead and checked it. I'm going to go ahead and turn it off, and I have a cup of hot water just ready. I'm going to go ahead and take them out, put the lids on, and then put them in the hot mug. Alrighty, so I went ahead and poured 4 dishes already. I'm going to go ahead and pour the last 2. And once I'm done pouring the last 2, then I'll go ahead and let them sit for about an hour. I will cover it, but not fully cover it, let it get some airflow, and then after that we'll get started with the experiment. Hello, I'm Vivian Fregoso, and this is our second lab for our third lab for our second week of microbiology. We are learning about our bacterial enumeration. So I went ahead and prepped our trays here, and I went ahead and filled up our tubes with 2.25 mLs of distilled water. Now I am adding in the yeast. So the yeast, it's about 0.25 mLs in each tube. So I'm going to go ahead and add it in here. All right. So I went ahead and added them in here. So now from our test tube, we are going to go ahead and get about, let's see here, we're going to get about 0.25 mLs of, 0.125 mLs of our yeast mixture with our distilled water. And we're going to put it into our dishes here. And I'm going to try to match them, you know, with like 1, 2, 3, 4, all the way to 6. And then we'll go ahead and put them in our incubation location. We'll check on them in about 24 hours. And then we're going to flip them upside down after 24 hours. So we'll definitely take pictures. There should be colonies located, or you should be able to see the little colonies. So in the meantime, right now, I'm going to go ahead and put about 4 drops of our mixture here into each one of our dishes. So I'm going to try to match them as best as I can. So let's see, looking for 6. So we have our 6 here. And they said about 4 drops or so. So let me go ahead and clean my pipette, move my yeast out of here. I'm going to go ahead and run it through the alcohol just to get it completely clean. That's about 4. For our 6, now I'm going to go ahead and cover it. So I'm going to take it from our number 5. There we go. Our 5 mixture. Now we're gonna do our number 4. One, two, three, 4. Here we go. And we're gonna do our number 3. One, two, three, 4. Back another one. And for our number 2. One, two, three, 4. Put that one back. And then lastly, we have our number 1. Two, three, 4. Perfect. All right, so now we're gonna go ahead and place this in our incubation period. I'm gonna clean out these test tubes with soap and water. And then I'm gonna go ahead and soak everything else in our bleach tub here for about 2 hours. I'll pour the alcohol down the drain, the yeast as well, dispose of it. So can't wait to see what the results are, and we'll see you back in about 24 hours for the picture. Well, we're gonna flip it over in about 24 hours, so we'll see you in about 3 days. All right, thank you.